Researchers use streak plates to obtain isolated colonies of target organisms. This is a well-formed streak plate of the bacteria Legionella pneumophila. Notice how the colonies are well isolated. In the lab, when making a streak plate, always start by sterilizing your gloves and your workbench with 70% ethanol. Wipe down your work surface and then light a Bunsen burner. Here we have 2 agar plates, a Sharpie to label them, 2 pieces of parafilm, our inoculating loop, and some sterile wooden dowels. Label the bottom of the agar plate, locating the 12 o'clock circle. Starting there, label your initials, the date, and the organism you are streaking with. In our case, we're using Pseudomonas aeruginosa, which is abbreviated by PA01. We have a blank and a streak plate. Now that the plates are labeled, we can place the blank aside. We are going to use a sterile wooden dowel to scrape a small amount of the target organism, Pseudomonas aeruginosa. Now touch the wooden dowel to the surface of the agar in a memorable spot. Typically, above the 12 o'clock circle is a great spot. Our bacterial stock is very concentrated, so this spot will contain hundreds of thousands of colonies. Now take your inoculating loop and flame it. We want to sterilize the loop until it glows bright red. Remember that we need to cool down the loop before we can touch the surface of the agar, as to not kill any bacteria we just plated. Once the loop is cooled, return to the spot where the colonies were placed. Drag your inoculating loop over the upper right quadrant of the plate. Replace the lid and flame your loop. Once your loop is cooled, take your plate and gently turn it a 4th circle. We will now drag the inoculating loop through the next region. The point is to dilute the concentrated stock of bacteria that we had placed on the surface to achieve pure, isolated colonies. Again, we will use the inoculating loop to zigzag through the remaining 2 quadrants. Each time we will flame the loop intermittently and replace the lid on the agar to maintain sterility. Finally, we will flame the inoculating loop and set it aside. The streak pattern shown here is called the 4-streak method, where there are 4 individual streaks in different directions. As demonstrated in the lab, we started at position X and streaked in 4 different directions and flamed intermittently to dilute the concentrated bacterial stock. Once you have finished making your streak plate, parafilm the agar plate shut and leave it to incubate in the 37 degrees Celsius incubator for 24 hours. It is important to incubate agar plates upside down so accumulated condensation does not fall on the bacteria's surface and instead falls on the lid of the agar plate. A successful streak plate is shown below, where isolated colonies are clearly and distinctly formed, and the blank plate is free of any contamination. Notice that the bacterial colonies are uniform and homogeneous in size, shape, and color. This should serve as a check that your isolated streak plate is a pure culture. Streak plates with contamination will yield plates that are not uniform and have bacterial colonies of different sizes, shapes, and colors. These streak plates are unsuccessful streak plates because no colonies were successfully isolated and there seems to be contamination on both the blanks and the streak plates.